Good morning, everyone, and thank you for joining us. I would like to thank Jean McAvoy and Julie Carson for their significant help and cooperation. The program today offers one CEU for pesticide license renewal and one CEU for certified crop advisors. The title of the seminar is Citrus Disease Trends We Should Heed, Phytophthora, HLB, and Leprosis. The speaker is Dr. Ozgur Batuman with UF, working at the HLB-IFAS Center. In his talk, he will remind you to continue paying attention to other pathogens that can attack already HLB-weakened trees, Phytophthora or Leprosis. Each can substantially reduce productivity of a citrus tree, but when they are co-infecting, it can be fatal. He will also let you know that some control measures may not be effective anymore due to HLB-induced complications. As you know, we have Phytophthora, we have HLB in Florida, but we don't have Leprosis yet, but it's knocking on our door. It's in Mexico. It's all yours, Dr. Batuman.
All right, perfect. First of all, thank you very much for all who found time and join us this fine morning. I really appreciate coming and listening to what I have to say. But at the same time, I should acknowledge our Manji, Julie, and Jean for putting together this seminar series and inviting me to give a presentation on some of the subjects that I really care and try to work as much as I can. So as title suggests here, that I will be talking about the 3 different diseases, and Manji introduced it very well, that Phytophthora, it's creeping up on us, and HLB is in the center of this title. HLB is not a new disease, but make an analogy today's situation where we are with the COVID-19 virus is being more deadly to those that who has underlying conditions. So let's think about the HLB throughout this presentation as one of the serious underlying conditions like diabetics or heart condition that our citrus trees have. And imagine that Phytophthora as a cancer coming on top, or the leprosis is coming as a COVID-19. So if you use this analogy, and probably you will understand what I wanted to give this presentation and what I want you to pay attention to. With that, I'm going to start my presentation by reminding that, again, HLB is out there, and it is a disease that you are all very much familiar with its symptoms, biology, and the damage that's causing to our citrus growers in Florida. So I'm not going to go and talk about all this subject titles that I put it here, but again, the HLB is our underlying condition, but it doesn't mean by any means that we give up on working HLB. And of course, we are continuing working on HLB, and it's more, harder than ever. Many scientists around the world really trying to come up with the out-of-the-box solution that might help our citrus industry here in Florida. Just to summarize what we have out there, and you can see here the control mainly can be divided in HLB disease control itself. It's a disease in terms of like bacterial agent that's causing this HLB disease, and also the vector, the psyllid vector that transmit the disease. And you will notice in this slide that most of the control strategies highlighted here with the red either failed providing effective control, or they are still being evaluated to see if they can provide some reliable control for HLB. So HLB control is still problematic, but as you can all agree, that psyllid control is still feasible, and it must be continued to reduce the inoculum titer and prevent the young trees getting infected with this HLB disease. Of course, we are all tuned and waiting to hear whether peptides that we've been talking about these days, and CRISPR, new technology, or other molecular techniques might provide some sort of control, hopefully in near future. But overall, if someone asks me if there is any control for HLB that they can implement today, so I would say no, there is no one single control. So it's, in other words, there is no silver bullet. So if you want to control HLB, you have to consider multiple aspects of integrated pest management strategy using different techniques. As I was mentioning, that there are many researchers, including many here in IFAS, working or collaborating with different researchers and scientists around the world to come up with the novel strategies. And I'm happy to announce one of the latest NIFA project was approved. It's titled here, Collaborative Approach to Discover, Develop, and Commercialize Therapies for HLB. This project is not necessarily a new project, but most of you probably familiar with the collaboration that CRDF started with the private company Bayer. Now this with this federal grant, we are expanding on our previous efforts to come up with a real practical solution that soon I'm hoping and very encouraged that we will be able to provide some solution. And in this new project, we are not only looking at or screening for available compound libraries in this one of the largest pharmaceutical companies in the world, the Bayer has, for curative effect, and also try to see whether we can find a compound that will give us preventive, control opportunities. So we are working on this, continuing to working, and we have very good results. And hopefully in 6 months from now, the same organizers of this seminars will be inviting me to give a little progress on that project. And following that project, we are still continuing to use modern techniques like CRISPR, RNAi to come up with the reliable and applicable practical solution for HLB control. And one of them probably you heard is CRISPR gene editing, or using the citrus tristeza virus as a vector to modify citrus defense system, where citrus becomes more tolerant to HLB or resistant. It is in the early stages of the project, but we have very good results, and hopefully someday I will be able to talk more about this. And the last project that I wanted to remind you that we are still working on coming up a new delivery system where we'll allow our growers to apply therapeutics material to the citrus that will allow us to deliver them to the citrus vasculature more effectively than it would with the spraying or drenching. And I'm of course not going to go in details, but I cannot stop myself not to talk about a little bit about this technology that we are envisioning here. So it's basically using the citrus trunk to deliver this chemistry instead of doing foliar spray or the soil drenching, where we are creating small holes and introducing chemicals passively, as we say here, needle-assisted trunk infusion, or with a little bit of pressure, it's like trunk injection to introduce chemicals to the citrus and distribute that chemistry homogeneously in the citrus, as you can see here in the small pictures with the dye experiments that we conducted. Of course, the ultimate goal with that is that this method will work and will be automated. It's imagine that like we are coming up with the trunk injection system that you will be able to install on one of your existing farm vehicle and go out and trunk inject, as you will, many trees to deliver chemicals more effectively and more targetedly control HLB, and maybe in the future for psyllid and other citrus diseases. So again, there is no silver bullet yet for HLB, and I'm not a believer of silver bullets, and it will never be a silver bullet. So I'm sorry to be straightforward with that, but again, I'm a strong believer of integrated pest management, and I do believe that it's our best option to continue to stay productive with the citrus in Florida if we continue to use multiple IPM strategies together, and it is possible, and most of you are very, very successful using IPM, and those are like you all do monitoring for pests, taking care of them, reducing inoculum sources, killing the psyllids, using this individual protective cover, etc., etc. So all this together is the aspects of integrated pest management, rather than relying on the pesticide that will solve the problem. But one thing that we are all focusing these days is I just wanted to talk a little bit more, just for the sake of the next pathogen that I will be talking. It's actually is taking advantage of this HLB created stress and disabling citrus biology that is causing more damage when they are infected with more than one pathogen. So, which is that HLB causing the not only foliar and leaf decay, but also impacting the root growth and reducing the root mass that they can produce throughout the year. So it is very critical to remember that HLB is changing almost all well-designed cyclical events. For instance, flushing, flowering, and root flushes, etc., etc. Put it in disarray. So every management strategies that we may have can become ineffective because of this irrational growth of citrus, or the panic mode of citrus trees to stay alive and produce some fruit. So with that, I will switch to, before that, sorry, I just wanted to reiterate the IPM strategy. So IPM, as you can see here, I tried to put in this pyramid mode that it's consists of multiple culture to protect our crops with different strategies. And of course, chemicals is part of the IPM strategy, but it's alone should not be relied on. And in the meantime, until we will have more tolerant or resistant varieties to control HLB, we really have to consider using IPM strategy to control HLB or reduce the damage of HLB. And I'm transitioning to other pathogen. This pathogen is, most of you familiar with, is caused by water-loving mold. Actually, it's not a fungus anymore, but Phytophthora name is the same, and it's causing severe diseases. Especially in recent years, their incidence is increasing throughout the state. So I thought I will make this connection with the HLB-weakened citrus, and the increasing Phytophthora incidence are linked somehow. So you will pay attention when you are trying to control this disease. So just a little overview on Phytophthora, as you all know, can attack different parts of the growing citrus tissues, including fruits, roots, and young plants trunk as the bark continuing to grow. So it can attack and cause disease, as you know. For instance, foot rot, trunks, and limbs, as you can see here, one representative picture. And if it attacks to the root, it causes root rot. And if it attacks the fruit, it causes a brown rot. So altogether, Phytophthora really by itself can be very destructive pathogen. And with the HLB in the background, it's just adding the problem in higher magnitude. So we have to really think about Phytophthora, not only Phytophthora today I'm going to talk, but in general, other pests and disease that can, increase the severity of HLB associated decline of the trees. So Phytophthora that we have here, two different species, but they are all causing one way or another diseases that we are all familiar. They have little differences in their biology and the molecular backgrounds, but namely we have Phytophthora nicotiani, that common cause of foot rot and the root rot. And the other one is Phytophthora palmivora, causes mainly brown rot of fruit and root rot in poorly drained soils with a high water table. So I'm not going to go in details of these pathogens. As I mentioned, there are the water-loving and the soil-borne pathogen. So all the propagules that we talk about Phytophthora and the infecting bodies actually can stay in the soil for a long time until the conditions are right. And there is a growing root, young roots. So please pay attention. This growing young roots are the fuel for the Phytophthora to come up and infect the citrus trees. And again, with the HLB, root cycle disrupted. So we always have some sort of trying to grow new roots. Hence, the Phytophthora that is on the soil with the right condition here, the warm the condition and the wet condition that will need for germinating this swimming spores to come and find this growing roots and infect, or the fruits that hanging low on the canopy of trees, they can splash the inoculated with the rain or other irrigation practices and cause infection and perpetuates this infection cycles more than ever before the HLB era. So Phytophthora, that's why is still causing a problem, and it's ever increasing to cause problems. So one of the disease that causes, I mentioned, foot rot, and as you can see here, some varieties actually very susceptible to this. And this disease kills the bark and sometimes even girdles entire trunk of the trees and suffocates the plants by blocking water and carbohydrate transport and leading the death of the trees. And it's relatively much easier to control this Phytophthora by using resistant varieties, especially when you are putting new resets in historically problematic Phytophthora problematic areas by choosing tolerant or resistant rootstocks. Of course, avoiding wounding nearby the soil or having the graft put on more than 6 inches above the soil will prevent this problem coming to your grow. The Phytophthora root rot and other way of infecting plant is attacking the root, as I mentioned. Is the root rot causes this Phytophthora from the soil and impairs water and nutrient uptakes by attacking and killing this mainly fibrous growing roots. And also by losing the roots, plant is losing the storage material for next production cycle, which are carbohydrates and other molecules that will be used for producing fruit next year. So it is very important that we protect our new trees, especially roots, by considering good drainage and taking care of the irrigation in where you will not allow creating the conditions conducive for a root rot. And sometimes I know that the nurseries are doing a great job and they do whatever they can to control this in their nurseries, but sometimes there will be incidences that it will escape and it will not be noticed and brought into your grow with the new nursery material. So please pay attention what you are buying and what you are planting. And afterwards, of course, monitor, or before, monitor and see how much Phytophthora load you have, the inoculum, titer, as we say, or you are familiar with the propagule count that Syngenta is still providing this service, I'm assuming, for free. So you should be looking at the inoculum load before making decision for choosing rootstock and planting your resets. Of course, IFAS has very detailed control strategies, and I'm not going to go in details because this control strategy is still effective. But because of the number of reasons that I've just told, that the HLB caused disruption of the biology of tree, so maybe the way that we apply this, control strategies or timing of application should be modified because of the HLB created this, unsynchronized root flush and growth pattern that we have. So I'm not going to go in details here. With this existing control strategies, it is still effective. It's worst case scenario. You have to really continue to do what you are doing at. So before going to details, I just wanted to finish up with another disease that's caused by Phytophthora, which it is brown rot, and usually attacks the early varieties. And when the conditions are right, you will see this brown leather decay on fruits on the tree, and later can produce white fungal growth and with the very distinctive pungent odor. So you can say that there is a Phytophthora involved or brown rot is happening. And then if unnoticed, this can be, continue to infect other fruits after the post-harvest storage condition. Of course, there are a number of ways that we can prevent or reduce this disease by eliminating the fruit under the trees. Again, it is impossible almost to do that because of the HLB created fruit drop, so it's almost not feasible to get rid of all the fruits. But there are ways, like for instance, rising the skirt of the tree to allow the air movement and drying the soil and the ambient temperature in the canopy in a way that Phytophthora brown rot cannot cause problem. And also we can use preventive sprays consist of phosphites or copper. They are effective to protect our almost getting ready to harvest crop. And sometimes when the rains are longer than usual, maybe additional application of this preventive material is required. So now I'm going to share some of the nice data that Mr. Tyler with Syngenta shared with me, and actually we had very nice discussion about it. That him and I want to bring up to your attention that you see there is a inoculum buildup in our soils throughout the states. As Syngenta collects samples from different parts of the Florida to see how much propagules sitting in the soil are citrus growers. And as you can see these graphs, I'm showing only the part of the 2013 till 2020. Every year it's kind of, there is a trend of increasing propagules on the soil. And at the same time, from those samples, the amount of roots mass that they are recovering, it's substantially reduced. In other words, inoculum is building and number of roots produced by citrus is reducing. As you can imagine that the propagule and root ratio is increased, hence there is a huge risk of getting more disease and more damage from the Phytophthora. Again, remember, HLB is disrupting the root flushes, so it's always producing a material for Phytophthora to infect and reproduce and become more problematic. And here is another data that I'm sharing from Mr. Tyler. It's from 2020. As you can see, the root mass, it's like almost 1/4 of the root that citrus tree used to produce before HLB exists these days, okay? And look at the propagule counts in last year. And even in areas where Phytophthora historically never been such a problem, as you can see, the number of propagule counts are increased. So we have to definitely rethink how to use available control strategies to maximize the benefit from this knowledge that we may share and use. So again, the main theme here is HLB reduces efficacy of Phytophthora management, and a possible reason for these are I listed here for you not to misunderstand. I'm not suggesting that any available material in the past is ineffective anymore, but just the way that we use or the timing of usage must be modified to make it effective again, because as if look like the effect that or the control that we used to get, we are not able to get it because HLB is change the tree response to all matters of stress, including the fertilizers that you are putting. It's not uptaking because of the lack of the growing roots and the response the HLB-weakened tree to relocate energies to other parts to consume whatever you are giving to. Okay, so again, remember that propagule counts are increasing and the root masses that the tree can produce are reducing. So the inoculum pressure is constantly building as your tree is weakened by HLB. And you are doing all you can to keep your trees happy and healthy. But unfortunately, even though you are providing, for instance, fertilizer, it's not uptaking because there is no root to take it. Okay. Or the vasculature, it's not as functional as used to be to translocate that material to suffering parts. Anyway, so the root cycles are not synchronized anymore. So as I said, and also disease production is not cycled anymore with this. So there's always some root and infection cycle going on. That's why I am suggesting that improving fungicide time in, and maybe number of application or the dose of application should be modified in a way to protect as much as root that we can. And I know it's not cheap proposition, but if we can remember the tree physiology and biology, and remember that after every spring or the shoot flush, we have a root flush coming. And imagining that the Phytophthora is increasing when the root flushes is coming, it is giving us beautiful windows to pick and choose which the root that will be more important for us to protect and get in turn a better crop. So there is a way to do that. And I believe all growers can monitor the situation in their grow by looking back their historical data and Phytophthora incidences, tune up and modify this Phytophthora control in much better and effective way and continue to take care of Phytophthora. So again, the suggested propagule counts is more than 10 to 20, but maybe we need to have a look at this threshold that was set 30 years ago, come up maybe earlier or with the less propagule count will warrant us to take care of the situation before it gets out of your hand and unmanageable and causing stress. So we have to think about this, okay? So I'm going to switch gear to another pathogen. As Manju suggested, no alert, we don't have this disease yet. It's called citrus leprosis disease. So I'm going to try to introduce you what is this disease and how it looks like and how it's transmitted, etc., for you to be watch out, because as Manju suggested, it is spreading very fast in other parts of the world where the citrus grown. And I'm going to mention about some of the places, and you will understand why we have to be watching out this. So my hope that you will get familiar with the symptoms, not the details of what type of viruses and how many of them and, you know, etc., but get familiar with the symptoms to recognize when you are scouting your, grows, you will be able to tell if this is canker or it's maybe leprosis or something else. So let me give you a little bit background about this disease. It's not a new disease. It's actually the first reported from Florida a long time ago. But because of the, the freezes and extensive sulfur application, it's somehow disappeared from Florida in late 60s. And since then, nobody's seen this disease in Florida. So today, we don't have leprosis in USA. However, the leprosis is considered one of the most important emerging citrus diseases. And you are going to ask why. Look at the spread of this disease in recent years. So it started from a bunch of countries in the South America and quickly, spread northwise to the Central America countries listed, some of them here. And in 2008-10, it's totally established in the south part of the Mexico. So it's just like our neighbors have it, and we may have it again. So it is, that's why I would call an approaching threat to the Florida citrus, and we need to be on the lookout for sure. So that's why I wanted to introduce you this disease. It's not a systemic disease. It's caused by the virus that cannot move from inoculated part to the other part of the plants, but it's vectored by the flat mites or false spider mites called brevipalpus mites. And we do have it here, not only in Florida, every citrus growing areas in the United States. So the vector is here, and the disease in the past was here, and it is a potential threat to our industry. So it is caused by the virus, as I said, and this number of viruses causing leprosis disease, and there are up to 12 now, and they are grouped because of their similar biology and the symptoms that they are causing, and considered to be one citrus leprosis causing viruses. And as I mentioned, I'm not going to go to in details what are these viruses, and probably you will hear sometimes the C-type, N-type. These are just to say where the virus is infecting and accumulating in the cell. So it's not very important for now. But later in the future, if God forbid and this disease comes, of course, it will be important aspects to learn and remember. And again, this disease can infect many rootstock and scions. And as I listed here, you will be saying, oh, some of them we do grow in Florida. So it's most of the citrus that we grow here, they are susceptible to disease. So we don't have called resistant or tolerant citrus available yet. But the overall message here, sweet oranges and mandarins are more affected from this leprosis alone. Imagine that we have HLB again, and this disease is coming. and its effect will be much severe than anything that we are seeing in the South American countries that is happening. It's not only citrus, by the way, is they are infecting. They can also infect other ornamental plants like hibiscus. I see here, I show here with the symptoms even, and the Dieffenbachia and other, dumpkin. Maybe some of you know that it's very common ornamental plants around us, so they can be hosts for this mite and the virus. So if you have around your grow these ornamentals, maybe it's imperative for you to go and look for this disease. Again, this leprosis disease is not systemic or transmitted by the seeds. And so it's very local infection. In other words, this diseased lesion, as you can see here, cannot jump from here to another unless picked up by the mite and fed here. Or if you take that disease part, bud, and use for grafting, this virus cannot spread systemically, okay? But the mites are very effective transmitting it, and we have it everywhere. They can colonize most species of citrus and other plants, as I mentioned. So you have to be careful with the flat mites population, not allowing to increase in large numbers to allow them to pick and transmit this virus disease. So again, vectored only by flat mites, and we do have it. And all stages of mites, including larval, nymph, and adult stages, can pick up this virus from infected tissue while they are feeding, and they carry this virus throughout their life. And when they move to younger, softer, or better tissue, they transmit this virus throughout their life. So it is important to remember that mites are transmitting this. I'm going to go and give some, information about the symptoms, how they look on stems, leaves, and fruit. So overall, this virus can infect and localize leaves, fruits, and stems. Comes up as lesions. But at the same time, also they can lower the fruit quality and cause fruit drop. Although it's just external cosmetic damage, but it can really cause the fruit drop, the premature leaf drop, decrease the foliar area, and branch death. And with the combination with HLB, of course, can cause tree death. And maybe some of you would remember the scaly bark disease that it used to call, for this leprosis disease in Florida, because of the bark lesions or damage caused on the stem, rightfully so, scales-like looking lesions. But most characteristic symptom will be for you on noticing on fruit or leaves, these large, relatively large chlorotic lesions. And usually you will be able to see this, as you can see here, I'm sure it's very clear. There is a feeding wound where the mite was feeding and introducing this virus, and the virus is moving around that in the focus area to other part of the leaf. So its chlorotic lesions will be very, very distinctive to other, known symptoms that we can see. And here are another samples on the upper part of the leaves of relatively large, chlorotic virus lesion. It can be pale green color too, very yellow, and sometimes they can produce gums, as you can see here. And from the underneath, looks like this sometimes. And here is one very nice example, actually. And this large chlorotic lesion that I mentioned is not necessarily large or always is distinctive. As you can see in the early stages, it can start small maybe, then it expands. And as it expands and the tissue become older, produce necrosis or dead areas and cause gummosis as well. And here, one lesion on the upper part of the leaf, at the same leaf from the down. As you can see, this lesion visible from both sides of the leaf. And here is a very severe incidence of chlorotic symptoms on leaves. I hope some of these photographs will stay with you, and you will be looking around to see if you may have this disease. So as I was mentioning, there are a number of different viruses causing, and one of them is called N-type because it accumulates in the nucleus of the cell. Usually the symptom of that type of virus, again, it's with the yellow halo lesions, but it's more chlorotic in the middle, as you can see, and the lesions are relatively a little smaller than the previous one, as you can compare this with this, but nevertheless caused by virus, and it's called leprosis disease and transmitted with the same mite, and it can cause disease on leaves, fruits, and stems. So leprosis can cause this lesions on twigs, stems, on the green part of the growing part of the plants. In early stages, they can be isolated, and then they can coalesce or become more corky or slow off even in advanced stages. So it is really sometimes can be confused or misdiagnosed by other disease like cancer, but I'm hoping to show you some nuances and will help you to distinguish between cancer and this. So another a slide showing new early stages of this stem lesions on the young growing stems. And on fruit, again, is the similar chlorotic lesions. In early stages, this chlorotic lesions in the middle is greener, as I try to depict here, green. And as it's advanced, it becomes more necrotic and brown, as you can see here. And in the more advanced and severe cases, these lesions can become sunken and necrotic, as you can see here. But as you can see here, it's causing, as we say, like rusted nail head looking lesions. So these are the chlorotic lesions in different stage of development. So again, early symptoms on the fruit and more advanced symptoms on the fruit with more necrotic lesions expanded in this infected area. Again, another type, N-type, can cause much smaller lesions on fruit as well. And actually, this can be easily confused with the canker or citrus black spot disease. So we have to be really, really be careful before concluding or deciding what to do. So one example here, I think it's summarizing my fear. It's really, really very difficult to sometimes distinguish between canker and this leprosis. Not only canker, also sometimes even the feeding damage of pests can cause. For instance, sting bugs are increasing now. Sting bug damage actually will mimic exactly what I showed in the first slide, that the feeding necrotic focus and the chlorotic ring around it will look like leprosis. But anyway, long story short, I want to just remind you that the canker, as you all know, that has this rampant and rough edges. So when you touch it, you can feel that it's like a ramped area. It's like a volcanic eruption occurred, and you can feel the sandy feeling. But in leprosis, one is more smooth, and you will not have that feeling. So it's more, smooth, and you will not feel any the sandy or rough feeling that you would have canker. But again, visually, they are really, really very similar. So we have to be, again, very, very careful. There are control strategies for leprosis, so we are not in the dark, but I just wanted to emphasize two things. We don't have in Florida, but it doesn't mean that it will arrive. So preventing leprosis to re-entering Florida is much more easier than finding here and trying to eradicate or eliminate. So that's why I know that you are doing a great job not bringing any material from outside without proper ways. So if we can keep this leprosis out of our groves, I think we will be better off. Again, we have bigger battles going on in our groves, HLB, Phytophthora, and we don't need this leprosis. And it is controlled in other countries, mainly by controlling this vector, flat mites that I talked about. So it's God forbidden, if it comes to Florida, our first, line of defense will be controlling this flat mites better than ever. But again, preventing will help us much, much better than any other control measures that we need to adopt and use. And with that, I just wanted to tell you that I know it's too much information to, take it and remember maybe, but please do remember that FDEX and us, the IFAS faculty, and all extension agents and specialists, we are all here to help you to A, diagnose the symptom, and B, come up with some advice or help to eliminate this disease in your growth. So please do not hesitate to reach out, any of us. Personally, you're always welcome to call, email, text. I will be happy to receive samples and come and look and try to help as much as I can. And we are all in the same boat on this problem. So let's continue to work together and help each other to prevent this disease becoming more severe in our citrus grow. With that, I'm not going to continue with this because I really wanted to leave a couple minutes for discussion, and I like to get your question. But before that, I really wanted to thank my especially grower collaborators and as well as the funding sources that allowed me to work some of this diseases that I introduced or the projects that I introduced. So I really want to thank to them and the many talented people that helping me to work on this important project. So with that, I'm going to stop and thank you for your attention. And I will be happy to take your question if you have any for me.
Thank you, Dr. Batuman. If you have a question, raise your hand and Jean can unmute you and you can ask your question.
And if not today, feel free to email me, my contact information here. Email me, call me. I'm always available and be happy to, try to answer your question